Boa tarde a todos. Sejam todos bem-vindos a mais uma palestra do 5º Simpósio de Micoses, mania de falar micoses, mas de infecções emergentes e reemergentes no Brasil. Hoje vamos a ter o honor de escutar a palestra do professor Nosanchuck, que proviene da Albert Einstein College of Medicine. O professor Nosanchuck se formou na Cornell University, na College of Arts and Science. Recebeu seu diploma médico pela Cornell University também e completou sua residência no Hospital de Nova York, que pertence à Cornell. Depois de várias bolsas de pesquisa, ele se alinhou na divisão de doenças infecciosas do Departamento de Medicina da Einstein e del Montefiore, que é um hospital associado ao Albert Einstein College of Medicine, e depois ingressou no corpo docente como instrutor em 1998. Atualmente, o Dr. Nosanchuck é reitor associado para o setor de educação médica. Ele é o que controla e faz toda a parte de entrevista e aceitação dos alunos de medicina, que lá nos Estados Unidos funciona um pouquinho diferente a como é o acesso aqui no Brasil. E ele é professor atualmente de medicina, fundamentalmente de infeccioso, tanto desde o ponto de vista microbiológico como imunológico. Atuou como reitor assistente em esses estudos focado fundamentalmente aos estudantes de medicina, e o professor Nosanchuck, a parte dessa área muito importante voltada à educação, ao contato direto com os alunos, é um cientista extraordinário, né? Então, ele recebeu o prêmio Samuel Rosen como o melhor professor pré-clínico de destaque da Universidade, o prêmio Harry Eagles por o melhor ensino de ciências básicas em 2016, e ele realiza um amplo serviço, ele presta serviço como médico para a Sociedade Americana de Doenças Infecciosas. Ele atua no conselho editorial de várias revistas científicas de relevância internacional, tem mais de 300 artigos publicados, vários capítulos de livro, tem financiamento do NIH e do Departamento de Defesa estadunidense. Ele é membro eleito da American Society for Clinical Investigation, da American Academy of Microbiology, do American College of Physicians and Infectious Diseases, da Society of America. Depois de se formar na Cornell University, Josh começou a trabalhar fundamentalmente em micologia, fundamentalmente com fungos como Histoplasma e Cryptococcus e Candida, e fundamentalmente estudando tanto tratamento como terapias imunológicas, como descrevendo os principais fatores de virulência desse fungo. Josh, it's a pleasure, an honor really to have you here. In the name of the Biofísica Institute from the Federal University of Rio de Janeiro, in my name, thank you very much to be here with us. And it's going to be a pleasure, of course, and we are going to learn so much listen to your presentation about Candida auris. Please feel free to start your presentation.
Okay, I'm going to share this. Let me know if it works. And
it is working.
Okay, and also, Susanna, if you can, if there are questions that come up, I can't see anything other than this screen through this program. So let me know if anybody has any urgent questions.
Okay, but feel free to go ahead with your presentation at the end.
Okay.
They are going to ask all the questions together.
Great. Okay. Well, I first want to make some disclosures in terms of where I've been supported. I have funding from the NIH and the Department of Defense, and The work that I'm going to be talking about today is in part funded by the NIH, and I'm going to talk a little bit about some product from a company that I'm part of. But first, I want to talk about Brazil for a few moments. And Brazil has been very important in my academic career. And I want to start with Professor Carlos Taborda, who's in Sao Paulo, and he came from Professor Travassos' lab, and he joined Arturo Casadoval's lab in 1997, where I was also a postdoc. And he really introduced me into many things about Brazil. And we've become very good friends, and we continue to collaborate. But the introduction to Brazil followed with meeting other people like Rosalie Zancope-Oliveira, who's at Fiocruz. And my first student was this guy here, Professor Alan Guimarães, who's in Niterói. And this is his wife, Mariana, who came also from UFRJ as a student. And after Alan, many other students came, including Professor Goides, Herbert, who's on the call here. And then I also got to meet many other people from UFRJ. And you can see here, Professor Susanna, and this is Alan again. And I also want to highlight that this is on a Saturday morning at 10:30. And when Susanna and these others were in Einstein, they were always here at the middle of the night, on weekends, and just always trying to learn something new. And they were just absolutely fabulous to have in my laboratory. Carlos Taborda also introduced me to Marcio Rodriguez, who's a professor at UFRJ, as well as Fiocruz, and now he's at the Fiocruz in Curitiba, and others as well. So this is Kildare, my friend, and this is Professor Niemrichter. You might think Leo is asleep here, but he's actually thinking about science. These are new experiments, and he's always thinking about great ideas. The other thing that's important with Professor Niemrichter is that he trained this person here, Danielle Zamith Miranda, who has been in my lab for the last 5 years. And much of the work that I'm presenting today comes from Daniel. But I would be remiss if I didn't say that there are so many more Brazilians. I saw Eliana a minute ago, who I've been able to work with over the years. It's just been an incredible journey, and I want to thank all of you who've been my friends and collaborators over these years, and I hope we can continue to work together. So with that, we're going to turn to Candida auris. So this is a very new organism in many ways. It was only described in 2009, and there are some that are older when people went back and looked in their collections, but really it was 2009 when we first started thinking about this fungus. And one of the reasons why it's so important is that most of the isolates that we deal with are either single drug resistant, multi-drug resistant, or pan resistant. The mortality rate is very high. On average, it's about 50%. This organism is incredibly resilient in the environment, and we're going to talk about that a little bit. It causes outbreaks. So a single hospital may not have any cases, and then all of a sudden you have 2, 5, 7 patients who have this fungus in your hospital. It's not easy to identify without high molecular methodologies. So this is a fungus that was surprising. Where did it come from all of a sudden? And it's a very scary fungus too. So this is a map showing you that it's on all continents except for Antarctica. So it's globally spread from that time in 2009 till today. It's everywhere. How come? Why? Is it climate change? Is it travel? There are many different possibilities. This is really the time frame in which you saw it. First in Japan, then in Korea, and again, they found some earlier isolates in Korea. For several years, there was very little found, and then starting in 2013, you started seeing it in India and the UK, and we saw it in the United States. And you can see here, as it goes from different colors, all of a sudden we found different clades. So here are 4 different clades of the fungus that have been found. And a couple of years ago, there was a single isolate that was found in Iran, which is thought to be a 5th clade. So where did they all come from? And why has this been so effective in terms of causing disease? In the United States, our experience was the first case was identified back in May of 2013. And you can see here, over a very short time, in a couple of months, we were having 40, 50 cases, and unfortunately, it's continued to go up. Why? In the United States, it's probably from travel, and people are coming and going all the time, similar to Brazil. People are coming and traveling everywhere around the world. Clearly slowed down with the pandemic, but there's still a lot of global movement. Just want to show you how this evolved in the US. So in the initial years, it was mostly in my state in New York and New Jersey. And then you see it starting to spread. And in 2018, it was required to notify if you found this in your laboratory. And then you see it continue to spread such that this past year, we have it on many different states. And these black dots are where we had it in one year, but not in the current year. And you can see how fairly spread it is across the country. And last year, we had about 1,000 cases, of which close to 300 were in my state, New York. It's important to realize, though, that even though we have a lot of Candida auris, it's still not very common. We mostly have in the United States still Candida albicans, followed by Candida glabrata, and it's less than 1% that we see with Candida auris. But it's still incredibly important for our healthcare systems. But what about Brazil? For the last several years, I've been talking to my friends and saying, do you have it there yet? Do you have it there yet? And I kept hearing no, no, no. Unfortunately, in December of 2020, there was a report from the area in Salvador where they found 2 patients that had this fungus on them. They were not sick with the fungus, it was colonizing them. And interestingly, Candida auris has been found in many patients with significant COVID-19. And we can talk a little bit about why as we move forward. And both of the patients, it looked like the strain came from South Asia. And when they looked at more patients, they found that more were also colonized with the same fungus. And now Brazil is very closely monitoring whether this is going to expand and disseminate. But so far, to my understanding, it really has not, which is really important. How do you identify it? It is a yeast. It replicates by budding. Typically, it's in singles. Some strains clump. You can see here up top this clumping. It's unclear what the relevance of the clumping is. Some people say that the ones that disseminate are more virulent. Others didn't show that. So it's still debatable whether clumping or not clumping is associated with virulence. Importantly, how you can also differentiate from Candida albicans, it does not form germ tubes. And it really doesn't form hyphae or pseudohyphae very commonly. In certain stress situations in the laboratory, we can make it form pseudohyphae, and sometimes in tissue you can see pseudohyphae. It grows very well at different temperatures, which is important in terms of its resilience. So in the laboratory, we usually start with biochemicals. And this is a problem with this fungus because it's misidentified most often as Candida hemoloni. And early on, the automated systems weren't identifying it correctly. Now, systems like this one called Vitec2 works well, but that was not the case for the first several years. We typically use mass spec, which is something we're going to be talking about for our science later on too. But in our hospital and most of the hospitals in the US, and I know that Brazil has been at the forefront of using MALDI-TOF also for identification. It's a very effective way of doing it, but it is costly and it's limited in terms of the number of samples you can put in. You can also do sequencing, but that also takes time. So it's a delay in diagnosis, which is important because you want to treat these people as soon as possible. I just put this up so you can see how far removed Candida auris is from things like Candida glabrata and Candida albicans. It is very different than these commonly seen Candida species. So why are we, again, so interested in this? The high mortality rates. In the US, we usually see around 30%, but in some places, particularly those with under-resourced areas, the mortality rate is 75%. Who are at risk? People who are compromised. So people in intensive care units because they have lots of central lines and they're on antibiotics and antifungals. These people are at very high risk. this fungus is the first fungus to be identified as a global threat by the CDC. And the European equivalent also is called this a global threat. What do we see in patients with Candida auris? Well, I told you that oftentimes it's just colonizing, so it's on the skin. But the clinical presentation of somebody with the disease is very nonspecific. You can see it in the bloodstream, which is probably the most common. We also see it in a lot of wounds, both surgical and non-surgical wounds. So in red are the patients that I've dealt with, and also with colonization. I have not seen here anybody with a heart disease or with disease in the brain, but it's something that is reported. And why the association with Candida auris and COVID? Well, because those are people in ICUs. Those are people who are getting antibiotics and antifungals in some places. So they fit the risk criteria for having this as a opportunistic infection. So colonization, this pathogen doesn't like to go away. And in fact, in New York, when we were looking at clearance, only about 1 in 10 people were able to get this fungus off of the skin. And if it's still on your skin, that means you can spread it to other people. And it can also be on surfaces. So it can be on doorknobs and bed frames. It's just really hard to get rid of. When we've looked at this, the majority of people were colonized before they had a systemic disease. So colonization is very, very important. And drug resistance. So this is an older slide, and you can see even in 2016, most of these were either resistant to one class or two classes. And even back then, there were some pan-resistant isolates. The clades are really variable in terms of their susceptibility. In Southeast Asia, almost all are resistant to fluconazole, and about half are resistant to amphotericin, a very powerful antifungal. In East Asia, they're mostly susceptible. Since we in the United States have a mix of these clades, we have about 90% that are resistant to azole now, 3% that are resistant to the polines, 5% resistant to echinocandins, and in 2019, we started seeing pan-resistant isolates. In fact, just recently, over the summer, there were cases of people with pan-resistant isolates. And so where were they? They were both in DC, Washington, DC, and in Texas. Very separate, no association between these patients. So in 2 different areas, we had these 5 pan-resistant isolates. Very, very concerning. How do you get drug resistance? Some of it is very similar to what we know from other Candida. So mutations in the sterol synthesis pathway, transport pumps or efflux pumps, regulating those to either keep things from coming in or pumping out the antifungal. And then there are mutations in what's called the hotspot area for echinocandins that have been determined. And then biofilm is something that's very important. You can see here these beautiful pictures. They're aggregated. There's not a lot in many of these of this extracellular matrix that you see in this species right here, but they are very tightly put together. And so biofilm is another very important factor for resistance in Candida auris. I want to make sure that you know that echinocandins are what we typically give for initial therapy. And that's a challenge in some poor resource places because they're expensive and they're only given intravenously. But we are going to talk about echinocandins as I move forward into the science part because we've used this drug to look at responses in the fungus. So not a focus of what I want to talk about, but we in our lab have looked at treatment of this fungus and really looking at it more from a skin approach in terms of decolonization. So using nanotherapeutics, a nanoparticle that releases nitric oxide, we were able to show that you could eradicate these different strains both in a planktonic as well as in a biofilm form. So this therapeutic approach to eradicate the fungus from the skin may be a way of reducing risk to that patient from subsequently developing disease, as well as spreading it to other people who may develop it. So now we're going to get to some of the work that we've done here. And the question really was, what makes this fungus so resistant? And one of the things that we were thinking about was this central metabolism process. Why is this organism able to adapt to so many different environments? Candida albicans is only in humans. It doesn't really live on surfaces. It doesn't live in water, and it really is a unique human pathogen. But Candida auris is able to go and survive in so many different areas. So it must be more metabolically malleable. And so we took our approach of using multi-omics to assess what was different between this fungus and Candida albicans. And I want to point out that this work was done with Daniel Zamith, who came, as I told you, from UFRJ. Ernesto Nakayasu, he is from Sao Paulo originally. He did his PhD work there. And then along with Marcio Rodriguez, Leonardo Nimrichter, and Alan Guimaras, we've been working very closely on these topics. So what we did is we took one of our strains from my hospital that was highly resistant to fluconazole. Basically, we could not give more than a milligram of fluconazole to get it into the solution, but even with that, it was still growing. And it's also resistant to caspofungin, one of the echinocandins. The other isolate we used was susceptible to fluconazole, caspofungin, as well as to the pollines. And we took a classic Candida albicans strain that we've used for other research in our lab and compared them. So I'm not going to go through all of these different findings because we have a lot to cover today, but I want you to look at a high level, and you can absolutely see that there are significant differences in regulation between the Candida auris strains and the Candida albicans strains in these various areas. So up here we have things like the TCA cycle, we have pyruvate metabolism, fatty acid metabolism, highly upregulated in Candida auris. And some other areas where we have ribosomal activity, gluconeogenesis, much more elevated in Candida albicans relative to Candida auris. And you can see that there also are differences between these two strains of Candida auris as well. And again, I don't want to take you through all of these things, but just notice that these blocks that have a solid box around them, there is a significant difference between the Candida albicans and that resistant strain of Candida auris. And the majority of these boxes are showing you that there is a significant difference between them. And what I really want to highlight is that Candida albicans, as I showed in the heat map, has more abundant processes in gluconeogenesis, phagosomes, whereas Candida auris is really very active with the TCA cycle, lipid and amino acid metabolism. When we looked at ergosterol, and ergosterol is an essential component of the cell membrane and regulates many of the homeostatic processes of the cell, you can see here too that there were significant differences in some of the processes between the Candida albicans and the Candida auris in many of these different steps toward the formation of ergosterol. And some of the metabolites, for example, farnesol is very important in regulating the hyphal switch in Candida albicans, and you can see that farnesol was very low in Candida auris, which does not have any morphogenic switches. So here's the take home from the ergosterol pathways, which was 11 of 19 of these synthesis enzymes were much more abundant in the resistant Candida auris compared to Candida albicans. So very dynamic in terms of its change. When we looked more at lipid biosynthesis, you can again see marked differences between the Candida albicans and the Candida auris strains. And again, for certain areas, you had very different regulation of the susceptible and resistant. In areas with these longer lipids as well as saturated fatty acids being significantly up in the resistant compared to the susceptible isolate. So we found that triglycerides and lipids, especially the polyunsaturated fatty acids, were higher in Candida albicans, but the lysophospholipids were significantly upregulated in Candida auris resistant strain. So we needed to step back and say, what does this mean? Well, it meant that we could generate these incredibly complex heat maps and flow diagrams that would allow us to start figuring out what molecular processes were different in Candida auris. And when we looked at this in terms of trying to solve drug resistance, we found that it was not a single thing, but it was multifactorial. And the differences were really broad, from carbon utilization to sphingolipids to the cell wall formation and efflux pumps. And here's an example of one of the efflux pumps, CDR1, where you see that the drug-resistant Candida auris was markedly upregulated relative to the susceptible one, as well as compared to Candida albicans. So this allows us to then start dissecting areas for further work, and we're starting to think about which genes to look at and which pathways to follow. One of the really interesting things in fungal biology over the past about 15 years has been the discovery and analysis of fungal extracellular vesicles. And this really began when Marcio Rodriguez was on a sabbatical at Einstein in Arturo Casadoval's lab when he discovered this in Cryptococcus neoformans. And since Marcio's discovery, we found, and this is only a short list now, of different fungi that produced extracellular vesicles. And these vesicles are really interesting because they have proteins, pigments, lipids, RNAs, and many of these are associated with virulence. In fact, many studies, including a large number that have been done in Brazil, have shown that in Cryptococcus, for example, the polysaccharide that Susanna has studied extensively is packaged into these extracellular vesicles. And these vesicles have dynamic responses with macrophages in terms of regulating all sorts of processes, from the release of cytokines to killing within these host effector cells. Extracellular vesicles can change macrophage polarization. They can change virulence interactions, not just with Galeria, but now Leonardo Niemeyer has done beautiful work in mice too. And the dynamic of response is really incredibly regulated. And we've shown this most clearly with antibody work, showing that just binding of antibody to the surface of the fungus can dramatically change the loading and release of these extracellular vesicles. So we thought that Candida auris was very likely to release extracellular vesicles, and they probably had a role in promoting or regulating disease. And so we started to look at this and looking at the molecular profile of extracellular vesicles from our Candida auris as well as Candida albicans as a comparative point. And I want to show you that this work just came out in August, so very new data. And the high level is that these EVs do have immunoregulatory properties and that they're not the same as Candida albicans. So I'm going to take you through some of this work here. And so you can see here that these are transmission EMs of extracellular vesicles from Candida auris, and they look nearly identical to those from Candida albicans. And interestingly, in the drug-resistant strain of Candida auris, you see that these vesicles are larger. Both the small population and the larger population are bigger than in Candida auris susceptible strains or in the Candida albicans situation. So again, very complex heat maps. I want to show again things like the TCA cycle are very different in Candida auris compared to Candida albicans. And to show you that loading and release is really highly regulated, and I'm going to show you some other data later on. Again, the EVs the whole cells compared to the EVs are very different in terms of the quantity of certain products in terms of what was most upregulated. So things like this heat shock protein 12P, the ligand binding protein, was significantly upregulated in the whole cells of both Candida auris strains with differences between them. And it's not significantly increased at all in the extracellular vesicles, whereas some of these manoproteins were markedly upregulated, but not seen in the whole cells. So a very dynamic process and different between the Candida albicans and the Candida auris. When we looked at what was abundant in terms of the proteins, we found about a third of them were differentially abundant. And again, I've talked about the TCA before, so I'll put that one in here. And you can see that the TCA was one of these areas where there was much more activity in Candida auris EVs compared to the Candida albicans. When we looked at lipids, we saw also very dynamic differences, and some of them were really interesting, like the ceramide here, where the quantity in the extracellular vesicle of this virulent strain was markedly increased relative to the cells, and it was somewhat elevated and more than what was seen in Candida albicans and the EVs. Again, showing that this is really a highly trafficked process and highly regulated. And looking at these other lipids, you can see that there were variations in the amounts that were put into the vesicles in the different isolates. So again, highly regulated, and are some of these clues that might be biologically active in terms of host regulation to increase pathogenesis. We also looked at RNAs, and RNAs had previously been shown by Liz Alves and Hosanna Puccia and others in things like Paracoccidioides and Cryptococcus. And yes, we too saw a fair number of RNAs, mostly non-coding RNAs. And you can see that there were some differences in quantity with certain types of RNAs between the albicans and the Candida auris. And if I pull up what some of the major ones are, you can see that they were very different in terms of the major ones, and also the changes were in different directions between the two. So again, highly regulated and differentially regulated between these species and between these strains. We do a lot of pathogenesis work, and we are starting to do more with Candida auris, but this is some early work where we were looking at attachment and interactions. And you can see when we added extracellular vesicles to these epithelial monolayers, we had significantly increased sticking of this virulent strain. So it's possible that these vesicles enhance the interaction between the Candida auris and tissue. We did do phagocytosis and killing, and so far with the epithelial layers and with some macrophages, we did not see differences. However, with dendritic cells, we did see differential regulation of some markers on the cells, as well as some differential activity in certain cytokines, like here's IL-6. And you can see that as with Candida albicans, giving these extracellular vesicles increase the amount of this cytokine, and in contrast, the vesicles decrease the expression of TGF-beta. So we did see regulation in dendritic cells. So far with our extracellular vesicles, what we found is that, yes, Candida auris produces and releases these incredibly biologically active structures, and that the content in terms of protein, lipids, and RNAs were really different from Candida albicans, and there were differences between the Candida auris strains. And importantly, EV biogenesis is a highly regulated process. It's not a random activity. We found that the extracellular vesicles increase the adhesion of the Candida auris to epithelium, which was a very interesting finding. And we saw that they were able to activate these bone marrow-derived macrophages, dendritic cells, but we did not see changes in phagocytosis by the macrophages. So I told you that antifungals were really important, and we don't know what happens in the presence of antifungals to Candida auris. And there's very little data about this in terms of gene differential regulation when exposed to conventional antifungals. So we chose to look at caspofungin, which is one of our standard echinocandins, and we wanted to look at what was happening in response to this antifungal in both the yeast cells and in extracellular vesicles. So we did transcriptional analysis, and we were focusing on RNA, and we figured that caspofungin would lead to some silencing of pathways in order to have some stress responses elucidated. And I want to just point out that this was just accepted last week, and it's hopefully something that you'll enjoy hearing about. So when we applied caspofungin, we found that there were some differential growth rates between our two strains that we used, and that the caspofungin at subinhibitory levels still did slow down the growth, even though it was at subinhibitory levels. And the subinhibitory levels actually reduced one of these strains' viability, even though it was at lower than the MIC. One thing that we also saw with these subinhibitory concentrations is that it caused the cells to aggregate, and some of the cells were smaller and some of the cells became a little elongated. So there were morphogenic shifts, which makes sense because the echinocandins regulate beta glucans, which are a major component of the cell wall. And one of the things that we saw with caspofungin is that it significantly increased the expression or the exposure of mannans on the cell surface. So here is without caspofungin, here is with caspofungin, so many more mannans are being expressed. And same with this other standard strain from the CDC, where you saw increased expression or increased access to mannans after treatment. And we did this also by flow cytometry, and we had similar results in terms of the expression of the mannans. One thing that we saw is that there were significant differences in the regulation of these transcripts, and that they were different. So in the MMC1, the resistant strain, we saw autophagy was upregulated. And autophagy in some cell systems has been shown to be associated with increased extracellular vesicle production and release. And we also saw that other things were downregulated, like purine metabolism and DNA replication. Over here, we saw that we had more spliceosome activity, and RNA processing was actually increased on the susceptible strain. Again, another heat map that's really complicated, but I just want you to see globally that there were changes when we gave caspofungin to both of these strains compared to the parent strain, and some of them were more differentially regulated. So here is MMC1, where you have this showing upregulation, and here you have downregulation in the setting of caspofungin, whereas in the susceptible, it was relatively downregulated to start with, and there was less of a change after the exposure to caspofungin. But very dynamic differences, particularly in things like our histones. You see here histone H2A, H2B, histone H3, histone H4, histone H3. So for some reason, histones are particularly altered in the setting of caspofungin. When we wanted to actually try and confirm some of our results, we turned to the mannoproteins again, and we saw that the mannosylation levels on the cell surface when we were doing our flow cytometry were indeed significantly increased with caspofungin. So it correlated with our findings by the transcriptomics. We also did proteomics, and again, very complicated. Interestingly, the multi-drug resistance transporter 1 was downregulated relative to in control situations. And that doesn't necessarily make sense, but biology is biology. And so what I also want to show you is that when we looked at our proteomics and our transcriptomics, they were fairly well matched with the changes in regulation. Again, so what? So caspofungin had significant morphological changes that took place, especially the aggregation that we saw, and it exposed a lot of mannans on the cell surface. We saw with transcriptomics that there were significant gene regulations in the synthesis of cell wall, which makes sense for echinocandin's target, as well as cell cycle and ribosomes. Our proteomics really confirmed what we saw with our transcriptomics. And basically, this is, again, a large platform of new information that now we can start targeting pathways and we can start targeting specific genes to explore these findings in more detail. So we talked about the cell and the responses, but what about the extracellular vesicles? Well, this is some ongoing work, and what we saw is that there were changes in the size and amounts of the vesicles in both of these strains when we applied them to caspofungin treatment. And we found that there were also distinct changes in the RNA that was packaged into these extracellular vesicles upon caspofungin treatment. And again, most of them were non-coding and so difficult to work with, but we have Liz Alves in Curitiba who's helping us decipher all of this work. But I want to again stress that what we saw were significant differences in terms of what we saw enriched in the cells and in the EVs, showing that this is a highly regulated process. It's not just if something is highly expressed in the cell, it's going to get dumped into the vesicle. So these are just various examples of where we saw differential regulation of these mRNAs being in the yeast cell versus packaged and released into extracellular vesicles. And this is just another way of looking at the distribution in terms of the different types of RNAs as well in these different cells and in these different structures. So what we saw here is that we had these genes, and again, I'm going to focus on the cell wall and plasma membrane, where we saw changes based on the expression, the exposure to caspofungin. The mRNAs and the extracellular vesicles were really associated with a global stress response that was expected by caspofungin, and that especially things like stress signaling and cell wall maintenance were really important. And again, the distinct regulation between the yeast cell and EVs from these distinct RNAs. This further is giving us insights into targets that we want to look at in the future as we start doing some molecular biology with this organism. So the last part I want to talk about is, are there other parts of the immune system that might be associated with pathogenesis in this organism? And so what we chose to look at was whether these extracellular vesicles had any influence on T cells. And what we found is that, yes, there were interesting activities where the extracellular vesicles inhibited T cell activation. They actually shut down activation. And you can see this through either looking at cytokines like interleukin-2 and interferon gamma, where giving the vesicles reduced the expression of these cytokines, or through FACS analysis, where when you usually apply extracellular vesicles from Candida albicans, you see this shift over to the left of activation, whereas giving vesicles from Candida auris does not cause an activation response. So we found a really interesting phenomenon, and this is the basis of the current grant that we now have and are beginning to study what factors in these vesicles are associated with this inhibitory response. I will point out that Candida auris also evades neutrophils, and this is done by my colleague in Madison, Janelle Nett, where they've looked at neutrophils and nets in terms of activation, as well as the changes in monosylation. And again, we were showing monosylation being something really important with echinocandins, and Janelle to date hasn't tried that with caspofungin to see if that's a change that happens with Candida auris, but it's something that I know she's now interested in doing. So I gave you a tremendous amount of data so far, and we have more, but I'm going to conclude here with what have we really learned about Candida auris. This is an incredibly surprising and highly pathogenic organism that has exploded on the global scene. It's particularly scary because of the high rates of drug resistance, which are associated with the very high mortality rates. The metabolic plasticity of this organism, and we showed you this through many different ways, with and without drug stress and with and without biofilm, it allows the fungus to survive. As we are uncovering these new virulence factors, using omics and other powerful methods, we have found these pathways, like that interesting ceramide I showed you, that we can study and figure out what mechanisms of pathogenesis are unique to this organism, and how can we better develop methods to prevent and treat the organism. And we also showed that this fungus can evade not just the neutrophils that Janelle showed, but that through these complex mechanisms, they can also subvert T cell biology. And the bottom line is we need to do more research. There are so many more things that need to be uncovered. So I have a lot of collaborators that I'd really like to thank, but primarily at Einstein, Danielle has been the driving force for this work. Leandro da Silva was in my laboratory for a year, and he's helped out on some of this work. He's back in Brazil right now. The T cell biology work is done with my collaborator, Fernando Macion. And I also really want to thank Susanna again for inviting me to be here. And I have some of my other collaborators here in Brazil, and I know I'm missing some. And I did not talk about my work with Attila, where we're looking at attenuation and virulence through doing growth assays with a serial dilution of antifungals. And that information is really interesting, and hopefully we'll have that out in the next 3 or 4 months. And with that, I will take questions. And my first question is, what are they drinking? I've never seen them drink water before, these guys.
I think they are sick. So thank you very much, Josh, for your incredible work. And because I think the work that you are doing now in auris are so important to understand what is different in that fungi. And I'm gonna open the microphone to Professor Norton Hayes, and he's gonna ask you some questions. He wrote it down on the chat, but maybe it's better if he opened the microphone.
Oh, thank you. Very nice work, Josh. I was curious about the sphingolipid metabolism. You mentioned it into your presentation. And I'm wondering if you have, you showed that there was an increase in ceramides.
Yes, and lysophospholipids also.
Yes, yeah. So I saw from my screen the ceramides were pretty short. Is that correct, or?
So there are a variety of ceramides that we found, but most of them were 18 carbons. So not really short, but
okay. No, I didn't see really. I was surprised, but anyway, I was wondering about the ceramides you showed, but what about other phosphosphingolipids like IPC and other molecules that could be going into this pathway that are shown to be involved in the pathogenesis of other fungi?
So I do not profess to be a lipid biologist. I'm really lucky that Leonardo Niemrichter and Daniel Zamith, my research associate, are lipidologists. So I don't want to mislead you with some of the information that I'm going to present, but we are still analyzing some of the findings in terms of trying to get better lipidomics. We're working with Ernesto Nakayasu, and he's at the Pacific Northwest National Lab, where they have incredible omics capacity, and we're trying to develop more effective methods for doing these lipidomics, because right now they're fairly complicated and we need a fair amount of materials. What we're really trying to do is to develop some better processes to look at the lipids in the vesicles, primarily to determine what might be associated with that immunosuppressive effect. And so if you have suggestions of ones that we should really keep our eye out for, we would love that, because so far we have a few different possibilities. The one ceramide we've tested so far was the one that I highlighted on the screen earlier. that was significantly upregulated in the extracellular vesicles, and that was the D18 ceramide, and that one did not suppress or alter the biology of that interaction. So our intent right now is to do a lot of lipid separation over the next couple of months, and start trying to determine, just based on some simple things like polarity and size, whether we could identify what might be the immunologically active lipids in the vesicles.
My last point is about the lysophospholipids you mentioned right now. Fungi, they don't make ether lipids. So the lysophospholipids you're talking about, they are acyl glycerols linked.
Yes.
And the phospholipase activity would be like a phospholipase A2, A1, and the polar head group would be choline.
So I think one of the interesting things about what you were just bringing up is what types of enzymes are prevalent in Candida auris, and that's an area which is not well studied. There aren't as many that have been seen, or as high quantity as are present in, for example, Candida parasilosis, which presents a lot of phospholipases and lipases. Candida parasilosis loves lipids. It's associated with one of its virulence characteristics, and this particular Candida auris doesn't seem to have quite the expression that parasilosis does, even though it's got very different biology in its portfolio of lipids. So, you know, we started studying this fungus in 2018, so it's still a fairly new organism in our hands. But it sounds like I should get Leo to go and sit with you and talk about different areas where we can start looking for activities that might further differentiate this pathogen from Candida albicans and parasilosis glabrata and others. But great questions. Thank you, sir. Thank you.
Thank you.
Now, Herbert, please.
Hi, Josh. Great job. Congratulations. Your talk. It's quite new for me, this Candida auris. So I have a first simple question. Do you need to work in biosafety level 3?
So that is a great question, Herbert, and one that has been discussed by many people. It is a BSL-2. Many people say BSL-2+. I don't really know what 2+ means, but we are very careful with this organism because it is so resilient and so easy to spread from person to person. And so we primarily work with it in the hood, whereas some of our other Candida that are BSL-2 will work out on the bench. So we do try and take extra precautions. Some laboratories have a single hood that they only use for Candida auris. So it is something to be concerned about, but it is not a BSL-3.
Okay. So about the interaction with the enriched cells, I saw your data about the vesicles, but did you try to do some interactions with the Candida auris? Maybe I missed this information. So
we have done some, but again, it's preliminary, and we want to do more. One of the things that we were waiting for, Herbert, was getting funding, and the funding for that study with Candida auris we just got about 25 days ago. So we're really excited to have the money now to do this work. And you know, right now Daniel Zamith is starting to make lots of vesicles, so we can do some of these host pathogen interactions. And we're also looking at some other virulence factors that are pretty interesting. And in order to do that, we're going to have to do more macrophage and dendritic cell work. So stay tuned, and hopefully I'll have more answers for you in the future.
And I have a suggestion for you about the suppression on T cells. Maybe it could be exhaustion. So maybe the vesicles are inducing PD-L1, And if you have because it's normal for parts of T cells express PD-1, so you can dial the production of interferon gamma in the other cytokines. And maybe it's a common mechanism for different pathogens now, so maybe it could be one of these.
No, that's a great point, and it's one of the areas that we're planning on looking at. I think the PDL pathway is really fascinating. I'm not sure if you remember, we were the first to look at that in fungi, and we showed that PDL1 blocking PDL1 was very beneficial in the setting of histoplasmosis. And other scenarios, it's very different, because my colleague then did it with tuberculosis, and when they blocked PDL1, they found that the tuberculosis was overwhelming and killed the mice very quickly. So these regulatory pathways are so interesting. And what was most interesting for me with the histoplasma and TB is that both of those organisms are intracellular, and they go into the phagosome, and they both regulate the VATPases and all sorts of things. So many of the biology is similar between those organisms, yet the outcome, and yes, one is a mycobacterium, one is a fungus, but the outcomes were incredibly different. But the
in Leishmania, we showed that if we use bulb seal, we enhance the lesion, but we control the parasite load. So we have a good and bad scenario at the same time. This is fascinating. Thank you so much, Josh. Congratulations for your work.
It's very nice to see you.
Yes, I have a question that is like opinion, maybe. Can we say that Candida auris is multi-resistant, or is more like not susceptible? Since unlike the bacteria became resistant because bacteria get in contact with a lot of drugs. But you can isolate Candida auris from the environment with all that multi-resistant characteristics. There is a study from an Indian group that they isolate Candida auris from a small island without people. So in the environment, Candida auris was multi-resistant. So is multi-resistant, or is simply not susceptible to drugs?
So I think that's an interesting question, and I don't know if it's one that can really be answered. I thought that study that you referred to was very interesting. Why would they find it just in the environment like that? But you could say, well, geez, it's similar to the Cryptococcus gattii that was found in Vancouver Island, and probably got it there through either a dolphin or some debris that washed from Australia. So the fact that they were able to find strains in the environment, and there aren't that many strains in the environment, and we need more strains in order to really study the emergence of these virulence characteristics. But the finding of something being susceptible or not, part of it is what are the mechanisms for those processes. If it's all a matter of a pump being generated and pumping it out, then that's just a biological phenomenon of the organism. Whereas if things are altered, then I think it's something that is a process within the cell that is being regulated in order to compensate for the presence of something. So that would be more of a not susceptible. I think many of these things are terms that have been used for a long time. And some people even use sensitive, but sensitive doesn't make sense because these are inanimate. They don't really. C. elegans has a neurology, but the fungi don't have neurons in them, so they can't be sensitive to something. I don't really have a good answer for you other than to say that if you're looking at intrinsic versus an adapted response, I would say that susceptibility is probably more adapted and resistance is an intrinsic.
For that reason, I told you, it's not a question, it's an opinion, because it's like, we can talk about that for hours with a beer and wine. So
sounds like a plan.
Yeah, maybe it's where Alan is right now. He told me, no, he has a meeting, and he told me to send you a hug because he cannot be here today. Eliana, do you want to ask a question, a comment to Josh?
No, I just want to congratulate Josh for the nice seminar. Lot of lipids that I love so much, and some monotheliation that is very important too. And I like this. I didn't know about that the role of the monotheliation in all this process. Thank you very much for your nice talk.
Oh, thank you, Eliana. And we have a lot of things to discover. I don't know about the regulation of the manoproteins, and I don't know which manoproteins specifically are upregulated, so we have to find out about that.
Yeah, nice work. Thank you again.
Thank you, Eliana. You stay well.
Our student Larissa said thank you to you for the presentation. And she's not a mycologist, she's a virologist, but she loved the talk. So just thank you very much. It's a pleasure. You are my friend, so it's twice a pleasure to see you here, see that you are healthy, all your family is fine. So this is a good sign. So I hope in a space, a little bit of time, you can be here again with us in Brazil. And I'm gonna pass the word to Anna to send you a lot of congratulations for your talk. And thank you very much to be here with us today.
Well, thank you for letting me show our work.
Dr. Josh, it was a great pleasure to have you here. Thank you so much for your incredible talk. And now I'm gonna end this section and say again, thank you. And everyone, it was very nice to have you all here. And I hope to see you next week. Have a nice weekend. Bye-bye for all.
There is a lot of students, but students are ashamed to ask questions in English. So. Are you in Einstein or at home?
This is my office.
The new office?
My dean office, yeah. It's very nice. It's big.
It's a big one.
Wow, it's really big.
It's big. It's got.
But it's only for you?
Yeah.
Wow.
Muito importante.
Importante.
But you